Thank you. The recording is in progress. So I will be talking indeed about the non-invasive methods to image the brain clearance system, and mainly also be focusing on the human brain clearance system. And just before we start, you have to know that my research group receives support from Philips. But maybe first, why am I, as a MR physicist in Leiden, interested in human brain clearance imaging? And that actually mainly comes from a specific patient population that we're studying in Leiden, which is the Dutch type CAA population. CAA stands for Cerebral amyloid angiopathy, in which amyloid gets stuck in the vessel wall and leads to this kind of bleedings. I hope you can see my mouse over here. Excuse me, you also see the white matter lesions. Over here you see some more lesions. And normally you see CAA in the elderly subject, frequently leading to also to dementia, also frequently seen in Alzheimer patients. But in this kind of genetic type, the Dutch type CAA, this kind of pathology already starts around 40 to 50 years old. Excuse me. And we think it's a clearance disease because also in when we look at the amyloid beta and the spinal CSF, we see that in the pre-symptomatic CAA patients, the amount of amyloid beta in the spinal CSF is lower than in age-matched controls. And we see that in symptomatic Dutch type CAA patients, these levels are even lower and much lower than in the controls. So that's why we're locally interested in the brain clearance system. And it was already very nicely introduced by Hélène that there are a little bit different viewpoints at the moment on how such a lymphatic system works, although in most aspects are similar between all these kind of different viewpoints. This is what also Hélène already mentioned to be the extended mixing model, in which we think that the CSF comes in along the artery, goes into the brain tissue, and there the ISF actually picks up waste products, and then these waste can either go out via the perivenous or the periarterial spaces, where we think that there's not so much net flow in the PVS, but there's more like a mixing effect going on, for example, driven by the cardiac respiration or the vasal motion cycle. Moreover, it's very important that if you're thinking about amyloid beta, that this is not the only way that the amyloid beta is cleared from the brain. Also, it can go directly across the blood-brain barrier. And so there are multiple pathways in which the brain is actually cleaning itself. Now, if you're thinking about how to study brain clearance, then most of the work in the last decade has been done with 2-photon microscopy, in which you really beautifully can see how all this waste is being transported across in these perivascular spaces to the brain surface and subsequently out of the brain. However, this needs a cranial window, you need tracer injection, and also frequently using ex vivo imaging. So certainly not doable in humans, and certainly not non-invasive. Of course, in PET, we can actually measure these kind of brain clearance mechanisms, as especially shown by De Leon et al. But of course, it's radiation, so we would rather use MRI. And like Hélène said, somewhat the golden standard at the moment for human brain clearance imaging are these intrathecal injections done by Kaya Ringstad in Oslo, which really provide this kind of beautiful images, how the contrast agent spreads through the CSF regions, goes into the brain tissue and subsequently washes out. For example, Gaia also showed that during a good night of sleep, you see that actually more contrast agent is cleared than if you are sleep deprived. But if you're looking at the spectrum, then I think there's really a lack of non-invasive methods to study the brain clearance in humans, because these intrathecal injections have, of course, clear drawbacks. So if we then look back at this kind of schematic, then one thing that you might notice is that actually quite a bit of this schematic involves water or water-like fluids, like the CSF flowing in, into the perivascular spaces, then the ISF, and then the outflow either along the veins or along the arteries. And this is of course very good news for MR, because MR is of course based on mainly based on imaging the protons in water. So this puts MRI really at the forefront of creating some non-invasive techniques. And then if we look at CSF or water and MRI, then actually we see that CSF is frequently seen as unwanted signal. In FLAIR, we try to really know all the CSF signal to better see lesions. And that's actually quite possible because the MR properties of CSF are so different, with T1s much longer and T2s much longer than normal brain tissue. So again, this gives great opportunities for MR to do some non-invasive measurements. And for example, one way, and that's one thing that I will be using more often in this presentation, if you're thinking at how this MRI signal depends on the echo time, you can see that the MR signal in the tissue, even in such a partial volume signal voxel, which has 95% of tissue, a little bit of blood and a little bit of CSF, you see that the tissue signal is decreasing very fast as a function of echo time. So if you just prolong your echo time long enough, you can see that even in a partial volume voxel with very little CSF, if you measure at very long echo times, you will only see the CSF. So this is a great way of synthesizing our MR signal to the CSF compartments. Finally, the other thing that we can do with MR is, of course, make use that CSF is flowing, and therefore you can also rely on phase contrast types of techniques. Now, we just had the neurofluid workshop in Rome, and actually one thing that was very clear again, that MRI has so many options, and that's of course why it's such a beautiful modality, that we can look at very different parts of this whole brain clearance pathway. But you have to realize none of these MR methods are actually imaging the complete clearance pathway. And fortunately, we can actually measure many of these subparts. So it's really putting this, the pieces of the puzzles together to try to understand this whole brain clearance system. So in this presentation, I will cover some of the available non-invasive MRI techniques, mainly focusing on human applications. And I will do this starting a little bit from large scale measurements to smaller scale, and starting from bottom side up, so starting in the neck. And then the first thing, and it has been done for already quite a long time, for example, by Noah Alperin, we can actually do real-time phase contrast imaging in the neck area, where we can nicely see the arteries and the veins, and we can do quantitative flow in the vessels. And if we then lower the vent, we actually start to see also the CSF flow over here. And then, of course, we can see how this is changing, how is the CSF flow depending on the cardiac cycle, on the respiratory cycle. And we can also confirm this kind of Monroe, sorry, I see there's a huge spelling error over here, Monroe-Kelly doctrine, actually 2 spelling errors, many, that actually says that actually because of the inflow of the arterial blood, actually the CSF is pushed out. Now, especially Noam Alperin nuanced this whole doctrine by saying that first of all, you have to look at the difference between the arterial inflow and the venous outflow. And even if you look at this difference, then you can still see that there's a little bit of difference with the CSF flow, and he contributes that to intracranial compliance. Now, this kind of same measuring of the CSF flow at the bottom of the brain was also used by the quite famous science publication by Foods and Laura Lewis in 2019, in which they actually used a very simple fMRI bold sequence like shown over here, but they actually made use that in the bottom slice, and the TR of the sequence was actually quite short, you have quite a bit of fresh inflow. And therefore, the fresh inflow of CSF gives this kind of very bright signal. And of course, the temporal resolution is quite high, so they can actually measure this with a high temporal resolution. And then you start to see this kind of patterns in which you see kind of CSF inflow into the brain And because you have a bold sequence, you can also measure the bold signal in the gray matter, and you can actually see that these two are a little bit related, probably to facial motion or neurovascular coupling. But then interestingly, they also studied these subjects in sleep. And the first thing that you notice is that during sleep, you really get these kind of slow wave alterations in bold signal. And then we see that, especially when you see that this bold signal is decreasing, that's when you see the kind of these peaks in the inflow of CSF into the brain. And you see also that this kind of peaks are much higher than in the awake state. So again, maybe confirming that the brain clearance is indeed enhanced during sleep. And this was then also looked at in the ADNI database, and there you also see that there was a relationship between this kind of CSF fluctuations in the foreventricle and in correlating to the gray matter bold signal, and they saw a difference in this kind of coupling between the two as a function of age. So going a little bit more to the tissue level, I want to show this kind of studies by the group in Finland, who use MR encephalography as proposed by Jürgen Hennig to really scan very fast whole brain signals. And then they can create these kind of great spectrums and see again how, for example, the respiratory frequency is influencing the CSF flow and the MR signal and the very low frequencies. Very beautiful studies showing, again, also a quite prominent role for the respiratory cycle in creating these kind of CSF motion. Really going to the movement of the brain tissue. This is a study done by the group of Jaco Zwanenburg in Utrecht, who actually used a dense sequence to really map how the brain tissue is actually deformed according to the cardiac cycle. Luckily, these are not a true deformation. These deformation are enhanced 50 times to better show how the brain tissue is actually deforming during the cardiac cycle. And this can therefore be it can be one way of how the CSF signal can be pushed out of the brain, and therefore creating a kind of a clearance mechanism, like pushing the CSF and therefore the waste out of the brain. So now let's move a little bit more to the ventricle system, and especially also the choroid plexus. And the choroid plexus is, of course, a central part of the kind of the third circulation hypothesis, in which the CSF is being formed by the choroid plexus and then being pumped around and also being taken up in these arachnoid granulations. Now, I personally don't really like this kind of term CSF production, because if we think a little bit more about CSF, we know that actually 99% of the CSF is water. And actually, we know that water is a little bit more freely flowing through the brain than just being formed in the choroid plexus. So let's see what we can image. Now, as already being introduced, I come from a background of ASL, in which we can measure perfusion. And I think most of you know the technique, but just very briefly, in ASL, we invert the spins in the inflowing blood. We wait about 2 seconds and we acquire an image. This image is not only reflecting the inflowing of labeled blood, but if we subtract this image from a control scan in which we do not invert the inflowing blood, we can actually get a measure of the brain perfusion. Now, of course, you can also use this for measuring the perfusion in the choroid plexus, and it was done by the group of Dave Alsop, by Li Zhao, and Li Zhao actually used very many different labeling durations and post-labeling delay time, so you really can see the inflow of the labeled blood into the choroid plexus, and they really get very nice images where you can see that if you wait longer and label longer, you can actually see more and more clearly the posterior ventricles lighting up in the ASL perfusion scan, and that's of course where the choroid plexus is located. But interestingly, they fitted a kind of dual compartment model to their data. And the most interesting parameter is that they showed that they also fitted the T1 with which the label was decaying once it exchanged from the tissue, from the blood into the tissue or into the CSF. Because that was what they were showing in the choroid plexus, is that the T1 with which the label was decaying in the choroid plexus was actually much longer than the T1 that the label was decaying in the gray matter. And this is showing that probably quite a bit of the label has actually gone from the intravascular into the intraventricular CSF. So it has been exchanging. So this could be considered a measure for this kind of water exchange between the choroid plexus and the ventricle CSF. Now, before this, Jack Wells actually already measured something very similar in mice, in which he actually used this very long echo time ASL to only measure the ASL signal that is arriving in the CSF compartment. So on the left side, you see your short echo time ASL, so perfusion imaging, and on the right-hand side, you see the long echo time ASL, showing the arrival of the ASL label within the CSF compartments, nicely showing here the ventricles. Now, we like this study very much, and we try to replicate this in humans. And therefore, we actually had also a similar scheme with increasing labeling durations and post-labeling delays. We used a multi-echo 3D Grace readout and actually see that actually our echo times became as long as almost like 2 seconds. And this is all kind of measured at a typical resolution of ASL, so a low resolution. And this is published in this paper by Leonie. And these are the results. So this is the short echo time in which you see a kind of a normal perfusion scan. And then if we're looking at these longer echo times and notice that the scale is 15 times more lower than in the perfusion scan, you actually start to see a different component which arrives much later. And you also see it arrives a little bit more along the cortex. And that's already the surprising thing. And you do not only see it arriving in the posterior part of the ventricles, but we actually also saw the arrival of the label spins around the cortex. Now you can quantify this, showing the CBF, arterial transit time, and this kind of exchange parameter. It's important to remember that this is exchange, that this is the exchange of label water from the vascular compartment into the CSF. We don't know whether actually the same amount of water also immediately goes back into the vasculature. So whether it's kind of a net transport of water across the brain CSF, the blood CSF barrier, or whether there's exchange of water. To me, always this movie is much more convincing, so that's why I spent a little bit more time on this, where we see first the arrival of the label spins in the brain tissue, in the gray matter, and only later we see the arrival in the CSF, and then not only in the posterior part of the ventricles where the choroid plexus is, but you really see it all along the cortex. So also in the subarachnoid space around the brain, we can actually see exchange of label from the blood into the CSF compartments. And then, of course, which happens, I think, frequently to us, is that with MR, you think you have found something very unique and you're the first to see it, and then you look back into literature and see that other groups actually already has shown something similar. This is a publication in 2017 from a Japanese group who actually used 17 oxygen water MRI, and they actually also showed that if you inject this, you don't only see this 017 arriving in the choroid plexus, but also around the cortex. So it's very similar to our findings. Now, I really like this kind of exchange measurements, and there are quite a new family of techniques that are looking at water exchange between different compartments. And I want to name two. The first one is the T1 Palin, which actually may use this kind of flare principle in which you use an inversion pulse. And when the CSF is going through zero, you either give a +90 or a -90 kind of excitation pulse. So you do like similar like a subtraction technique as with ASL, but you compare the +90 and -90. And in this way, you can actually very nicely depict, again, these kind of exchange from the water from the choroid plexus into the ventricles. A very similar technique, and I won't go into detail, is the FEXI technique, which is more a diffusion-based technique. The main conclusions, brain clearance is a hot topic, and I really think that we see that a tendency to go from the basic research to human research. The intrathecal injection, I would still consider the gold standard, but it's very invasive. With MRI, we're getting a lot more possibilities, and I think also in the next ISMR, we see many more techniques being proposed. Water is still an excellent candidate to monitor it. I think we still don't really understand how water is exchanging between different compartments. As like, for example, we have shown that the water exchange does not exclusively happen at the choroid plexus, but also around the cortex. And with that, I would acknowledge the work of that Lydiane and Leonie did most of it. And I would like to thank you for your attention.